Morning to lecturers and friends. This video is about virus-like particle vaccine. We are from group 9 and here are the group members. Virus-like particles, or in short VLP, are molecules that mimic viruses but are not infectious because they does not contain any viral genetic material. They are a very effective way of creating vaccines against diseases such as HPV, hepatitis B, malaria, and more, as they are very similar to real viral molecules. Introducing a VLP into the body will trigger an immune response, but a person will not experience any symptoms of the virus. Once the body has had an immune response to the VLP, it will recognize the virus and prevent infection in the future, giving people immunity to that particular virus. Creating a VLP vaccine can be used bacterial, yeast, insect, or mammalian cells. Non-envelope and envelope VLP as a platform for antigen presentation and vector. The internal cavity of VLP accommodates exogenous oligonucleotides rendered as brown gene-like patches, drug molecule in red circle and small protein in green pentagram that can be delivered after the VLP get to the target organ or cells. The outer layer of the VLP displays foreign proteins that are fused in red triangle and covalently conjugated in yellow-green circle to the particles.
Next, manufacturing process of VLP. The manufacturing process generally consists of three main sections. They are upstream and downstream processing, followed by formulation. The first step is cloning the viral genes of interest. The viral proteins can be expressed in various platforms such as bacteria, yeast, insect, mammalian, and plant expression system. Once the cells expand in the bioreactor, it will be infected with virus and grow at room temperature. After that, it can be harvested using centrifugation. After harvesting and cell lysis, clarification step is performed to ensure removal of cell debris and aggregates. Nuclease treatment is done to further purify the process by degrading residual nucleic acids during incubation. After incubation is complete, nuclease enzyme must be cleared out using filtration or chromatography.
Moving on to the next step, which is the primary purifications, where ion exchange chromatography or ultracentrifugations are used. VLPs are commonly purified by ultracentrifugations over a calcium chloride, sucrose, or idisocianol gradient. Next is inactivations. Baculovirus must be removed from the final product through orthogonal downstream steps during purifications of VLPs. As a safety measure, some manufacturers will perform inactivations of baculoviruses prior to removal. Inactivation is done by chemical method using formalin or BPL. For polishing step, weak ion exchange chromatography resin have been used successfully. In this step, VLPs have passed through the column while residual baculovirus and DNA bind to the column. And lastly, for sterile filtrations and formulations, the final product is sterile filtered using a 0.22 micrometer filter. As a VLP-based vaccines are typically formulated in sucrose and twins detergents, some final formulation may contain amino acid, amorphous aluminum hydroxyphosphate, sulfate, and others. This is an example for analytical assay for VLP.
Let's take a look at the advantages of virus-like particle vaccine or VLP. Well, then the first advantage of a VLP vaccine is that it is not infectious, right? So VLPs lack viral genetic material. As a result of the lack of the viral genome, VLPs are incapable of infection or replication, thus preventing accidental reversion as has occurred with attenuated viruses. Secondly, better safety profile. As previously stated, the lack of viral genome material renders VLPs non-replicative and non-infectious. As a result, this improves their safety profile. VLPs are considered significantly safer than many other virally derived vaccines by avoiding attenuated virus reversion or incomplete inactivation. Thirdly, enhanced immunogenicity. VLPs provide high immunogenic response because of their high density display of epitopes, the capacity to present multiple proteins to the immune system, and their size. Moreover, VLPs are unlikely to need adjuvants to be highly immunogenic.
Next, we move on to the disadvantages of VLPs. First, the stability of VLPs. Generally, they are considered more stable compared to other subunit vaccines. However, when the environmental conditions change, especially during downstream processing, VLPs can become very unstable as they lack of genetic material of the virus. Next is challenges in assembly. The genetic fusion of the sequences of epitope into VLPs can sometimes be challenging as they may lose their self-assembly property or cause particle misfolding. Lastly, the complex processes are very costly and time-consuming, which lead to difficulty in industrial-scale production and requires several quality control efforts as various downstream processing steps may worsen the VLP's quality.
The absence of viral genomic material has improved safety during both production and administration, which is a major component for the virus-like particle vaccine safety. The earliest vaccine indicated for hepatitis B virus, which is obtained by extracting the inactivated hepatitis B surface antigen particles from the serum of HBV-positive individual.
Next is halal issues of vaccine-like particles. According to DG of Health and DG of Jakim press statement, vaccines do not contain harmful material to the human body. It also contains other components that are permissible to be used, and all vaccines used by the MOH are registered with DCA. And in this issue, the examples of vaccine-like particles is Agarics, Cervarix, Recombivix, HB, and also Gardasil, and their sources are permissible. So in this method, if it is made from halal, clean, and pure materials, it is permissible to be used.